Okay, we are going to cover Chapter 4, Extensions of Mendelian Genetics. And the goals of this chapter are to distinguish the different ways that alleles can interact to affect the phenotype, demonstrate how multiple alleles at a single gene can lead to the production of different phenotypes, apply Punnett square when assumptions of Mendelian genetics don't apply, use complementation tests to determine if independent mutations affect the same gene, and to use epistasis in order to order genes and understand what exactly is meant by epistasis. So the previous chapter we covered Mendel's work. So Gregor Mendel did a lot of fundamental studies in pea plants, and he was able to elucidate many of the rules of genetics. He also made a lot of assumptions about how things worked, and these were true for the pea plants that he was studying and the traits that he was studying, but in general, they don't always apply. So in this chapter, we're going to explore some situations that violate Mendel's assumption. So specifically, one allele is dominant to the other. So the heterozygote, in the case of the pea plants, always look the same, had the same trait as the homozygote dominant allele. We're also going to look at situations where there's more than two alleles per gene. How do you handle those situations? What happens if multiple genes control a character? So in the case of seed color, there was a single gene that controlled whether or not the color was green or yellow. Are there places where multiple genes interact together, so you have to worry about two genes at the same time? And then situations where each organism might not carry two alleles, and why that would be. So I'm going to break up this lecture into three parts. So part one will be variation in allelic number and their interactions. So what happens if you have more than one allele or more than two alleles? What happens if they interact in ways that aren't just dominance recessiveness? Part two will be situations where multiple genes control a character. How do you handle that? What about gene interaction and epistasis? And then finally, part three will be complementation studies and sex-linked traits.